Good morning, good afternoon, or good night to everybody. Sorry, my Zoom just restarted, so I hope everybody can see me and hear me, and I'm going to start again. Good morning, good afternoon, and welcome. It's my pleasure to virtually welcome you at the Joint Genome Institute for the 5th Thursday of the 2021 NeLLi Symposium. In the previous sessions, we heard about the origin of the eukaryotic cell, about the tree of life, about the role of novel lineages in biogeochemical cycles, and about cultivation of new lineages. And today we will learn about microbial symbiont of microeukaryotes. We will have two 20-minute presentations with Q&A. So feel free to type your questions in the Q&A during the presentations. They will be read at the end, I think. And we will also have 5 short talks without Q&A, but you will have the possibility to ask questions to presenters and continue the discussion afterward during the breakout session in the breakout room. Our moderator for today is Giulio Petroni. He obtained his PhD in protistology in 2000. Then he spent a postdoc at the Technical University of Munich at Professor Schleifer in microbiology. And he is now a full professor at the University of Pisa in zoology. And he works on bacterial symbionts in ciliates. So please, Giulio, take it from here and have a good session, everybody.
Thank you very much. It's a pleasure for me to introduce the first speaker, who is Filip Husnik from OIST in Japan. Dr. Husnik obtained his PhD in molecular cell biology and genetics at the Biology Center of the Czech Academy of Science in 2017. Then he worked in Killing's lab at the University of British Columbia in Canada as an EMBO postdoctoral fellow. He obtained Okinawa Institute for Science and Technology as an assistant professor in 2020. His research is focused on major evolutionary transitions, such as the origin of eukaryotic cells and its symbiotic organelles, mitochondria and plastids. His favorite model systems are symbiotic organisms, insect, protists, and marine microbes. Please.
Thank you for the introduction and good early morning from Okinawa. First, I'd like to thank the organizers for giving me the opportunity to talk about Protist Symbioses and to including this session during the meeting. And I think it's very important for when we discuss new lineages of life to also discuss how they interact with other microorganisms. And that's what I'll talk about today. So if you look at the model systems we use for symbiosis research, there's a very clear bias. And I actually worked on animal symbiosis as a PhD student, so I'm partly responsible for this bias as well. So most of our model systems are animals and animal bacterial symbiosis. So here on this image, I have 3 model systems I think are quite well known in the field. So the pea aphid, bobtail squid, and corals in general, or cnidarians. But these model systems don't span the diversity of eukaryotes. So on the left, you have the eukaryotic tree with the main lineages, and animals comprise only a really small fraction or small slice of this diversity. So by focusing on animals, we are partly biasing the general rules. So when we say something about symbiosis based on the animal models, we are potentially missing a lot of information from all other eukaryotes. And that's what I'll try to focus on today. So this is not a data talk. I'll mostly talk about our recent review where we looked at hundreds of papers describing symbiosis of microbial eukaryotes. And I'll I'll try to highlight these sometimes overlooked things from the protist symbiosis field. I'll first look at the broad diversity of the symbionts and their hosts. Then I'll show a few examples of the functional roles of these symbionts. I try to avoid model systems that will be presented later on, either as flash talks or in the main talk. And then I'll try to compare what we know about the single-celled hosts compared to the multicellular hosts, and where actually we potentially bias the symbiosis rules by mostly focusing on animals. So first, the diversity. So in our recent review, we did a broad search of all the literature we could find, and we mapped the diversity on the tree of eukaryotes. And what you can see from this tree is that that single-celled eukaryotes host an immense diversity of prokaryotes, both beneficial, commensal, harmful, and the taxonomy is of both the hosts and the symbionts is also extremely diverse. So it does make sense to look broadly at all eukaryotes when studying symbioses. And from this broad search, we could identify a few lineages that are particular common symbionts, especially proteobacteria like alpha proteobacteria, two lineages, Holosporaceae, Rickettsia, very common symbionts. Also Bacteroidetes, Cyanobacteria, Chlamydiales, several others. And we could also see that, unlike in animals, protist symbionts can inhabit pretty much any cellular compartment of the, or bacterial symbionts of protists can inhabit pretty much any cellular compartment of the host. So cytoplasm, host-derived vacuoles, they can be inside the nucleus, mitochondria, plastids, you name it, anywhere in the cell. And we could also see that ectosymbionts are also extremely common, especially in environments that are somewhat extreme for many organisms, so low oxygen, nutrient poor environments. And so all of this was mostly bacteria. Archaea are relatively understudied when it comes to symbiosis, and most archaeal symbionts of protists are, or all so far, are methanogens. And usually they scavenge hydrogen from the mitochondrion-derived organelle called hydrogenosome. When you look at the functional roles of these symbionts, one thing I would like to highlight first is that really quite often it's context dependent. So whether you call the symbiont a parasite, commensal, or a mutualist can depend on many factors, and very often it's actually not clear, and we don't even know if there's a function at all. And to illustrate this context-dependent plasticity, I'd like to highlight one group of symbioses we call farming or gardening with these two examples, where, for example, in dinoflagellates on the right, they have these structures on the outside of the cell where they keep cyanobacterial symbionts. So it's more like a greenhouse, so keeping vegetables in a greenhouse. And so the symbiosis is relatively loose. And this way, it's not always clear how like both the partners benefit or how the benefit changes over time. And even, I think even better examples are these Dictyostelium amoebae or slime mold that have Burkholderia symbionts that actually induce farming of other bacteria. But depending on the situation, depending on the context, they can actually be both mutualists and parasites, or they can decrease the host fitness. So just keep this in mind when I talk about the functions. And there's also an evolutionary layer of these symbioses. So in our review and in general, we see at least 3 different groups of symbioses. The first are the professional, or what we call professional symbionts. So relatively ancient lineages like the Rickettsia that have adapted to the symbiotic lifestyle of hundreds of millions years ago. And they move between hosts, and they are the ones who drive the relationship. And then we have the symbioses where we call them destined for extinction, where they stay in the host for thousands of years, millions of years, but eventually they would be replaced by something else. And in very rare cases, the symbiont can become integrated into the host. I'll mention one of these examples later on. So the first functional category are metabolic symbioses. I think there'll be a few examples, so I'm not going to talk about this much. So the symbionts can provide amino acids, B vitamins, soluble iron. They can photosynthesize, fix nitrogen, detoxify toxic molecules. They can remove hydrogen, so the methanogens, hydrogen scavengers. Then there are a few more overlooked categories or not that often studied. Probably very important: defense and competition. In some cases, the symbionts can help with the movement of the host. And of course, symbiosis includes parasites and pathogens. And one very clear thing about symbiosis of microbial eukaryotes, we're probably only scratching the surface when it comes to functions. So very likely there are many other unknown functions of the symbionts. I'll highlight a few studies from the overlooked functions. So first are defense and competition. The first example, so-called epixenosomes. So these are ectosymbionts on a ciliate. They're very microbial, and they actually defend their host from a predator. So they have like this long harpoon-shaped extrusion that can help the host to evade the predators. And another famous example are the R-bodies, or the killer trait of some ciliate. Again, a large proteinaceous structure that's involved in competition. One of the few examples of symbionts helping the host to move is Mixotricha. So it's a parabasylate from an Australian termite. It's one known example where spirochetes, so bacteria, long spiral-shaped, actually complement its flagella. So they cover the surface of the cell. There could be several hundred thousands of these bacterial cells, and they provide the host with a movement function. Another example is magnetotaxy, potentially also understudied, overlooked. So these are symbiontids. from usually marine sediments. And so they also have ectosymbionts. Their cells are covered by ectosymbionts. And these symbionts are actually magnetotactic bacteria that help the host to orient itself on the Earth's magnetic field, which could be beneficial to find the oxic and anoxic areas of marine sediments. And one example of a parasite, so bacterial parasites and pathogens of protists are, in my view, surprisingly understudied. We found this example with a colleague who was looking for large viruses, and he found this strange bacterium from the candidate phylum Dependentiae that actually targets the protist mitochondrion, completely remodels the mitochondrion, and then lyses the cell. Very likely there'll be many more examples like this. We just don't see them that often yet. So the last part of my talk is comparing the symbiosis outcomes in unicellular versus multicellular hosts. And I think this is really important because most of our symbiosis rules are based on animals, where the population genetics processes can be in some ways different. The symbiont transmission is by oocytes. There's often specialized tissue for housing the bacteria. Generation times can be very different. In protists, it could be hours. In animals, it could be months, years. And this influences the rate of symbiont replacement, the horizontal transfer opportunity, the diversity of the functions as well. For example, animals have lost metabolic pathways that many protist lineages still have. So I'll just show two specific examples of what I think are emerging model systems for this kind of research. One is a group of ciliates called, or the genus is called Euplotis. And in Euplotis, what has been shown that the symbioses are extremely dynamic. There's a massive turnover of symbiont replacements, which originally wasn't obvious because most of the symbionts come from the same genus. So they seemed like an ancestral symbiosis. But in fact, the host just selectively takes this bacterium from the environment and it replaces the older symbionts. And sometimes it takes a different one. So this has been shown in the animal systems as well, but it's even more obvious from the protist systems that symbiont replacements are the rule. That's how symbioses evolve. And the second example, very quickly introduce, is Polynella, so a Rhizarian amoeba. It has these two sausage-shaped cyanobacterial symbionts, and that's the one example of a symbiont becoming organelle with proteins or with massive protein import into the symbionts from the hosts. And so far, this hasn't been shown that clearly in the animal systems. So the protist system has really helped to push the field forward. So to sum up, I would say that Protist symbioses are often very context dependent. There are many more functions than just nutritional supplementation, as it might seem from the animal systems. And symbiosis outcomes in unicellular hosts are in many ways quite different from the multicellular hosts. So it is worth studying them. Since it's 1 month from our PhD application deadline, I would just like to mention, if someone's looking for a PhD on microbial symbiosis of eukaryotes, please get in touch with me or apply. And thank you for listening. I would like to also thank my co-authors of the review I presented, my lab for being great. I did not present any of our results today, and our funding bodies, and I'm happy to take any questions. Thank you. Hello,
Is there any question? I can see on the chat. I have a curiosity. Could you provide us some insight on dependency, the TM6 you mentioned before?
This was mostly work by our collaborator, but if you look at the data that are becoming available for dependency recently, in my view, they're quite specialized for protist symbioses or being protist pathogens with their metabolism extremely reduced, getting almost everything or depending on almost everything from their host. So I hope we'll be seeing more examples of dependency symbiosis of protists soon. Does that answer your question, or?
Yes, we have another question. How common are microbial symbionts in microeukaryotes? Are most protist lineages associated with symbionts?
So I would say you can find symbiosis in most protist lineages, but there are some that are more common hosts. Of course, there could be biases. We study larger cells more, or the larger cells could potentially be able to host more symbioses. Some lineages that are extremely understudied are heterotrophic flagellates, so predatory small cells, but they're understudied in general. I would say algae are quite understudied, single-celled algae, green, both green, red, but you can find bacterial or archaeal symbiosis in pretty much any protist lineage if you look closely.
We have another question. compared to insect symbionts, do protist symbionts also have strongly reduced genomes?
So yeah, I did not include a couple of slides on genome evolution because it would be probably too much, and I know there'll be a few slides on that probably in the next talks. Yes, there are examples of highly reduced genomes in protist symbionts, but not all of them have highly reduced genomes. It depends on the age of the symbiont, on its function. If the function is very complex, like helping with the defense, then the genome does not reduce as much. But the smallest bacterial genome from a protist is around 160. I don't remember the exact numbers. Kilobase pairs, and the smallest from insects are around 115,000 base pairs. So it's very close to the most reduced symbiont genomes, but we're probably not seeing all protist symbioses. So my guess is in the next few years, we'll find some that have as reduced genomes as the insect symbionts. But it's not a general rule. There are symbioses, some of the ectosymbionts have large genomes, or if the function is quite complex, the genome reduction depends on what the symbiont is doing and how old it is.
Okay, thank you very much. I don't know if Frederick would like to add anything, because I'm getting a little bit lost with the questions.
I can answer the remaining questions in the breakout rooms. I see there are several more.
Perfect. So we can go to the first of the short talk from Jean-Marie Volland. Dr. Volland is a scientist at the Laboratory for Research in Complex Systems and works at the Joint Genome Institute in the single-cell group. He uses classic and cutting-edge imaging approaches in conjunction with sequencing technologies to investigate unusual life strategies and symbiotic interaction in uncultivated bacteria in small eukaryotes. Please.
All right, thank you. I'm trying to, and hopefully it works now. So thank you. I will give you a brief overview of our project, where we establishing Zoothamnium Niveum as a model system for chemosynthetic symbiosis. And very briefly, because we are probably all convinced here already, symbiosis as defined as the living together of different species.
Sorry,
Marie, it looks like your presentation is not on. Could you double check?
Yeah, I can restart. For me, it's on, but the Zoom is making these funny things today. So let me try to share again. Yeah, try to share. Oh yeah, okay, no, sorry. Is that better?
Maybe we start with another short talk presenter, and then Jean-Marie, we have you at the end. Maybe restart the computer again or so.
Okay, so we can go on.
Can you see my slides now?
We can see your face, yes.
My face, but not my slides.
No,
let me try the sharing again.
Yes.
You have
now
it?
we see.
Okay,
yes.
Great. Okay, so I will start again. All right, so symbioses are interactions where sometimes the whole is more than the sum of its parts. And my slides are not moving. Oh, there it comes. And it's ubiquitous, and it's a driver of the evolution of species. And among the many reasons why partners form symbiotic association is food. And there are three categories of nutritional symbiosis: the heterotrophic symbiosis and the phototrophic symbiosis, where microbial symbionts use organics or photosynthetic carbon to feed their hosts. And in those categories, there are already a couple of good model systems that are established and used in many labs. But in the third category, chemosynthetic symbiosis, the microbial symbionts are autotrophs, and they are fixing carbon through chemosynthesis to feed their host. But in this category, there is unfortunately no good model system at all. Those symbioses are mostly known and studied from the deep sea, which is not an easy environment to reproduce in the lab. But fortunately, there are also systems that are ubiquitous, actually, in coastal shallow waters. And that's where we sample our favorite symbiosis, the Zoothamnium Niveum. It's a giant colonial ciliate, which can grow up to 2 centimeters long colonies. It is composed of hundreds of single-cell feeding cells called microzoids, and it is completely covered with a monolayer of a single species, a sulfur-oxidizing gamma protobacteria called Candidatus thiobios zoothamnicoli, which I think actually just got renamed. I have to check. We know already quite a bit about this symbiosis. It is, we know the developmental cycle, and we know that it transmits vertically the symbiont from parent colony to offsprings. The host has this behavior of contracting and expanding repeatedly. And as a byproduct, it brings the whole symbiotic population in the anoxic sulfidic boundary layer, providing access to hydrogen sulfide, which is the electron donor for the metabolism of the bacteria, and then expand again in oxic seawater, providing access to oxygen, which is the electron acceptor. In this context, the symbiont is very happy. It fixes a ton of carbon through chemosynthesis, and it will transfer some of this carbon to the host through direct transfer, and also a little bit through digestion of some of the symbionts. So it's a mutually beneficial symbiosis. It's a case of mutualism. We can collect and isolate the colony and make them release single cells, which can then be placed in flow-through mini aquaria or cultivation system. And when you provide them with hydrogen sulfide and seawater, they will settle and grow new colonies. And this cultivation system can be monitored and controlled for these physical chemical parameters. This is how it looks. Hopefully you can see the video. It's a time lapse of the first hours of growth of 3 colony growing from single cell. And you can appreciate the synchronous growth of the host colony and of the white bacterial coat that is covering the cells and the stalk. So our main goal is to develop an improved and accessible cultivation system for Zoothamnium niveum, which should be cheap, easy to produce and to use, reproducible and open source, in order to enable collaborative research on chemosynthetic symbiosis in a standardized way. So ultimately, there's going to be all the 3D files and SOP and tutorial videos on a website where you can then download the 3D models and print the pieces of the cultivation system. It's basically mostly printing a mold, which you will use to cast PDMS chambers. And then you assemble the PDMS body of the chamber with a standard glass slide and a frame and some sprues. And that's it. And you have your cultivation system for Zoothamnium niveum. You connect that with a continuous flow through of hydrogen sulfide, anaerobic solution of hydrogen sulfide and seawater. They got mixed homogeneously in this mixing channel, which we have simulated using CFD to make sure that it is reproducible and mixes correctly the 2 fluids. And within those chambers, you can see here on the picture, the Zoothamnium colony will grow attached to the plastic on the glass. We have first test round with a collaborator lab in the Caribbean that have been successfully isolating and growing the colony into the chambers. As you can see, after 7 days here, we have long colonies that start to themselves produce dispersive stage, and we then obtain a second generation, or they obtain a second generation of colonies, which we put under a scanning electron microscope to confirm that they were still covered with this monolayer of symbiont, and they were. So to summarize, I would say that we have simplified an existing system that now is composed of only 16 parts. You can build it yourself in any lab with basically just an oven and some vacuum pump to produce the PDMS chambers. The cost per chamber is only $20 compared to $240 previously, and it only takes 2 days to produce a few chambers, and it's also customizable. So, I will add that this cultivation system most likely can be adapted or directly used to grow different chemosynthetic symbioses from shallow waters. But for our system, I think we will contribute with a setup which will allow collaborative research. It's reproducible, it's low cost, it's going to be open source, and we hope it's going to contribute to the research effort on chemosynthetic symbioses and microbial symbiont of microeukaryotes. And with this, I'm done. Thank you.
Thank you very much, Jean-Marie. And let's go with the next talk by Sebastian Cristian Treitl from Charles University, Czech Republic. Dr. Treitl is a postdoctoral research in the Laboratory of Evolutionary Histology of Vladimir Hampel in Prague. Please.
Thank you. So, hello everyone. In the next few minutes, I will tell you a few words about Pelomyxa scheidi and its complex community of bacterial symbionts. But before we can start, let's introduce what is Pelomyxa. So, on the eukaryotic tree of life, Pelomyxa is part of the Amoebozoa group, where they are part of Archaeal, and they are closely related to free-living Mastigella and Mastigamoeba, but for other people more familiar is the genus Zoothamnium. They are giant amoebas. They are pretty big. Why they are so interesting to be studied? It's mainly because although they are so big, there's no proof for presence of the mitochondrium. So this is something which makes it very interesting for our laboratory. However, in order to study it, it's really complicated because it's a polyxenic culture. There are many other eukaryotes, not only bacteria. So it makes it difficult. And the way how we work on it, we use single-cell genomics and single-cell transcriptomics. And using these genomics and transcriptomics, we provide the first evidence for the presence of hydrogenosomes, or mitochondrial-related organelle. This part is already published, so it can be found, and you can read more about it. But the main takeaway is that we think that at least genetically, we provide evidence that there is a hydrogenosome which produces hydrogen and mainly acetate and succinate. However, to provide physical evidence like electron microscopy, we kind of struggle, mainly because the cell looks like this. So this introduces me to my main talk about the bacterial symbionts. As you can see, the cell, it's pretty much packed with bacterial symbionts. This is not something unheard from Pelomyxa. It was studied mainly in Pelomyxa palustris. It's a different species. In the 80s and 90s, it has been hypothesized that there are 3 symbionts at that time. They were based on morphology, but in 2017, a group of researchers managed to amplify the small subunits of these bacterial symbionts and prove that there are indeed 3 symbionts: a methanogenic archaeon, a delta proteobacteria, and an actinobacterium. So this is what was known, but since we studied Pelomyxa scheidi, we studied it with single-cell genomics. We obtained also the genomes of the bacterial symbionts. So we use bioinformatic tools to separate it. And initially we had 5 candidates. These are the 5 candidates which you can see here. using fluorescence in situ hybridization, we managed to show that these three of them, so an archaeon, a delta proteobacterium, and a firmicute, it's truly a symbiont. We show it by fish, so you can see clearly this is the metanogen, this is the firmicute, and this is the delta proteobacterium. And what is interesting, it's the fact that when you look layer by layer in the cell, you can actually see that the cell is pretty much packed with the bacteria, it's pretty packed with the firmicutes, and the metanogen seem to surround a round organelle. Initially, we were optimistic this might be the hydrogenosome, but as you probably noticed, this is too big, and it turns out that actually the metanogens do not surround the hydrogenosome, they surround a nucleus, and the reason why they surround it, it's unknown for us. But because we have the bacterial genomes and we have also the eukaryotic genomes, we are working on annotation. So this is preliminary data, and this is a flash talk. I cannot enter too much in details, but I would like to present our working hypothesis, how we think that this complex symbiotic system formed of Pelomyxa, which here will be represented by this blob. And these three are the symbionts. So here is the firmicute, this is the delta proteobacterium in red, and the metanogen in blue, and how this system would work together and interact. We think at the root of this system is the fact that Pelomyxa has the ability to aggregate cellulose and produce sugars. We also hypothesize that the sugars are taken up by the firmicute and the delta proteobacterium. They produce a lot of acetate, formate, and hydrogen. Of course, the sugar is used also by the host, producing acetate, succinate, and hydrogen. And the critical player here, it's of course the metanogen, which takes up all these byproducts, acetate, hydrogen, and scavenges it, uses it for its own metabolism to produce mainly methane. And we also know that the metanogen has the ability to fix nitrogen, producing ammonia, which might be used by everybody, but especially by the delta proteobacterium, which has a really high capacity of metabolic biosynthesis. It can synthesize pretty much all amino acids, and we think that somehow they share these amino acids between them. We also hypothesize that lactate is a product by the firmicute bacterium, but we are not 100% sure if this lactate is used by the delta proteobacterium or not at this stage. We are still looking into it. So pretty much this is our working model, how this complex system works together. And that being said, of course, I would like to thank our funding agencies, our collaborators, which help us with our electron microscopy, and of course, thank you.
Thank you very much. We can go on with the next talk by Victoria Jaco Reynolds, University of British Columbia. She's a PhD candidate in Professor Keeling's lab at the University of British Columbia, and her thesis work is about the character evolution of dinoflagellates. Please. We cannot hear you.
Ever since the discovery.
No, it's okay. Thank you.
So my name is Victoria Jaco Reynolds, and I'm a graduate student in the Keeling Lab at UBC, Canada, and I'm here to discuss the evolution of plastids and parasites, capturing the plastid metabolic dependency of the photoparasite Blastodymium. So ever since the discovery of plastid in the malaria-causing Plasmodium, there's gained lots of interest on how, in fact, can these intracellular obligate parasites evolve from free-living photosynthetic algae. So these apicomplexans are a part of a larger lineage called the myceliums of Aviolata. And sister to the apicomplexans are dinoflagellates. And within the dinoflagellates, there's a lot of diversity, but there also exists complex plastids and even plastid reduction and parasitism. So how does an agal cell shift from free living state to a parasitic form? And how does this transition impact the metabolic dependency on the plastid? So I'm interested in Denitrifying, a photoparasite of ecologically important marine copepods. So they're called photoparasites because they are actively photosynthetic within their translucent host. And to date, there's not many investigations on the combination of parasite and also being photosynthetic. So this system is extremely unique and really important. So I've been collecting plankton. I've been micro-dissecting 0.5 millimeter copepods for these Blastodynium parasites. And they come in several parasitic forms. You have initially the development of a trophomere, which will divide and provide gonomere. And it's the gonomere that keeps producing these sporocytes. And these sporocytes and trophomere will just fill up the whole gut of the copepod until the copepod can no longer feed and will eventually die. And this system is crazy because you also have a non-photosynthetic member of the Blastodyniales as well. And here are some more images of the non-photosynthetic member that I've collected in and around the Vancouver region. So when I find these Blastodynium parasites, they're not culturable, so I have to do plankton toes and do microdissections. And I've been collecting the cells using single cell isolation and creating transcriptomes for each of the individuals that I find and their diverse life stages. So far I've generated 14 Blastodynium transcriptomes, 3 pigmented photosynthetic individuals, and 5 non-pigmented individuals, non-photosynthetic. So I have diverse parasitic infective stages, and with this large data set, I've added my data to a large protein phylogenomic data set to characterize the phylogenetic position of these lineages. And so using 177 proteins, I've found that all my Blastodyniales are going in the Paradyniales. So of my photosynthetic individuals, they appear to be polyphyletic, and then following after the photosynthetic individuals, you have white Blastodynium, the non-photosynthetic, that seemingly perhaps lost photosynthesis once. Currently, I'm working on developing many single gene phylogenetic trees to characterize the plastids, so photosynthetic related pathways and housekeeping pathways as well. So far, I've found that the light harvesting chlorophyll acid binding protein is present in all of my green Blastodynium transcriptomes, and haven't found any at all in the non-photosynthetic. So what I aim to do is characterize the plastid metabolic dependency in different infective life stages of Blastodyniales, and compare this between the photosynthetic and non-photosynthetic members of this group. And I'm really excited to show you the results of this study. So for now, thank you to the Keeling Lab, Liz, Cooney, Elizabeth, Lily, and other Keeling Lab members. And this is our recent dive, and Patrick is just right behind me there. Thank you.
Thank you very much for the talk. And let's go with the next one, who is Emma George from University of British Columbia. Also, Emma George is a PhD candidate in Professor Killing's lab. And she studied interaction between protists, bacteria, and viruses.
Hello, everyone, and greetings from the Caribbean. I'm actually in the field currently, so this is a recorded flash talk. But I work on different bacterial symbionts of protists, and today I'll just focus on one study system, which is Cryptomonas. And so far, we've tallied up about 7 different genomes in this one single cell eukaryote. So there's huge diversity of protist symbionts, and we just put together this review recently summarizing all the taxonomy, functional role, and the localization of all these different kinds of prokaryotic symbionts in these major lineages of eukaryotes. And for some of the groups, there's been a lot of literature and a lot of data on different symbionts, but surprisingly for this group called Cryptomonas, the only symbiont data comes from microscopy studies. This is kind of surprising because Cryptomonas are known as a model system for secondary endosymbiosis because they obtain their plastid from red algae. And this group is kind of unique in the sense that it's also retained the red algal nucleus called a nucleomorph. So already Cryptomonas are very complex cells. They have 4 different genomes, including the mitochondria, the nucleus, the plastid, and this nucleomorph. So there's one freshwater Cryptomonas that's in the group of Cryptomonads that's even more complex because it has these bacterial symbionts, and the symbionts tend to have these capsid-like structures. This was described in the 1990s, but this culture has actually been going since the 1950s. So we were able to obtain it, and we did some transmission electron microscopy on it. And even 30 years later, this Cryptomonas culture still harbors bacterial symbionts, shown here. Plus, they have this capsid-like structure inside. So we did a lot of metagenomic sequencing on it, so using both Illumina and Nanopore to do short and long read hybrid assemblies. And we were able to assemble 2 different symbionts. They both come from the Rickettsia orders. And the first symbiont is Bacterial. It has a genome size of about 1.45 MB. Also encodes plasmid. The second symbiont is Microeukaryotes. I'm just showing one of the contigs here. It is assembled in about 5 contigs, and it's a bit larger. It's about 2 MB, and it also encodes a plasmid here. I won't go into too much detail into what they encode, but it's very canonical Rickettsia metabolism and also secretion systems. They are packed full of transposons and different mobile elements, as well as they encode several different toxin and antitoxin systems. So Microeukaryotes is known from several different hosts. It's been found in Ciliates, in green algae, Fusarium, possibly corals, and now Cryptomonas. And it belongs to the Rickettsia family. Where Bandiella has actually also been found in other Ciliates and is closely related to symbionts found in corals, the trichoplasts, so different animals. And it's in the Midichloriaceae family. This is actually the second time both of these 2 kinds of symbionts have been found in a single cell. They've actually been found together in Ciliates before. We were also able to assemble this really high coverage phage genome, and it has very similar GC content to Megaera. So we believe that it could be potentially infecting the Megaera symbiont. We still have a lot of research and different kinds of studies to do, including fluorescent in situ hybridization to determine the localization of these symbionts within the protist cell, and also to determine the exact host of this phage. So we have a real conglomeration of genomes. We have a host cell that already has 4 different kinds of genomes, plus 2 different bacterial symbionts and a phage infecting at least one of the symbionts. So there's a lot of competition going on here, And it's interesting to think about the selection because it's likely these symbionts are maybe competing for the host. And also we have a phage that's killing off at least one of the symbionts. We also have toxin anti-toxin systems at play. Both the bacterial symbionts encode them, and the phage, it has a toxin anti-toxin system as well. So these addiction molecules may be one reason that this culture has been going since the 50s and still retain these symbionts. And these Cryptomonas cultures, they actually have a generation time of about 5 days. So that leads to about 5,000 generations that we know of, at least, that has been in culture. So it's a really fascinating system, and I'm really excited to dig into it a bit more. And with that, I just want to thank the Keeling Lab, the funders, and also all the organizers for this great event.
Thank you very much for your talk. And let's go to the next one, who is Michele Giovannini from the University of Pisa. He is doing his PhD under my supervision and deal with the comparative and functional genomic of symbionts of ciliates. Please.
Thank you, Giulio. Today I will present the complete genome of Candidatus gromivella agglomerans, a novel intracellular holosporaceous parasite of Ciliate paramecium. First of all, I will start with a brief introduction about the disorder of bacteria. Holosporales are an order of alpha-protobacteria, which is composed only of obligate symbiotic bacteria, and they comprise many intracellular symbionts associated with protists, such as ciliates, but also with multicellular animal. However, some holosporales were found extracellularly in metazoan, especially in arthropods. About the phylogenomic overview, bacteria of holosporales were traditionally considered phylogenetically closely related to Rickettsiales, as can be seen on the left side of the tree, but modern analysis provide a different, more complex perspective in which holosporales are not sister group of Rickettsiales. The right side represents the currently most accepted position within with the holosporales phylogenetically close to Rhodospirillales. Very quickly, about invariability of holosporales, there are 4 families which are identified. 3 of them in green are found mainly in protists, while the 4 families in red is found only multicellular animals. Now I will show you the results of our work, and in this work we describe the symbiotic system formed by the Ciliate paramecium polycarium and the novel holosporales bacterium occupying its cytoplasm, frequently forming quite large aggregates, as you can see in the right image. We obtain, using space software, the complete genome assembly of Candidatus gromivella agglomerans formed by a single circular chromosome. And to our knowledge, this is the smallest reported genome among the order Holosporales. Phylogenomic analyses are in agreement with the 16 RNA gene. In detail, maximum likelihood and the Bayesian inference produce identical topology and place the Holosporaceae as a sister group of Cedimonadaceae with the Paracetibacteriaceae as early diverging. Gromivella was confidently assigned to Holosporaceae, specifically to the fast-evolving Holosporaceae represented by Cytomycobacter species and NeLLi, which are symbionts of diplonamids, rather than Holospora, which is another paramecium symbiont. In any case, relatively long branches leading to Gromivella and its relatives were observed, confirming the indication of faster sequence evolution. The comparative and function analysis show in the Venn chart on the left, the greater majority of the Gromivella cogs are shared with members of all 3 Holosporales families, And indeed, it represents a substantial subset of that of other members of the disorder. And consistently with the very small genome size of the symbiont, no particular peculiarity has been found. And the function of the only 5 cog is overall unclear, being derived from generic prediction and or not confirmed by direct sequencing comparison. While a more specific comparison was made between Gromoviella and all 3 small-sized genomes, Cytomycobacter species and H-Bacter. And so, as you can see in this bar plot, the genomic characteristics of differential of the different functional categories are very similar to each other. While, as shown in this representation, from a metabolic point of view, it's lacking. Indeed, the Gromoviella present a severely reduced metabolism with reduction in some pathways. Specific in this representation, red dashes, boxes, and lines are pathways which are lost in Gromoviella. For example, it lacks most key pathway involved in carbohydrates and energy metabolism. And according Gromoviella probably depends on importing many metabolites from its host. Notably, the full absence of the pathway such as glycolysis, Krebs cycle, and electron transport chain implies a direct dependence on host-synthesized ATP, probably thanks to the presence of 2 specific TLC nucleotide translocase. Furthermore, Gromoviella displays several systems of protein secretion, possible involved in the interaction with the parametric host. For example, both SEC and TAT system are present, and Gromoviella has a type 6 secretion system apparatus, including the majority of canonical components. And additionally, Gromoviella presents the components of type 2 secretion and type 4 pills. In conclusion, we obtain a new completed genome assembly of a symbiont of Parametrium polycarium, and phylogenomic analysis provide the consistent results, offering indication of the inner relationship within the fast-evolving Holosporaceae, namely supporting a closer relationship of Gromoviella with the symbionts of diplonymy, Cytomycobacter and H-Bacter, rather than with the Holospora. And finally, Gromoviella presents a severely reduced metabolism, which implies a strong dependence on Parametrium, and the presence of specific secretion system are probably related to a specialized parasitic lifestyle. I would like to thank all the people who collaborated in this work, and thank you for your attention.
Thank you very much for your presentation. And let's go to the last one from Jana Milucka from MPI Bremen, Germany. Dr. Milucka obtained her PhD in 2011 at the Department of Microbiology at the Max Planck Institute in Bremen on the dissimilatory sulfur metabolism coupled to anaerobic oxidation of methane. She's currently a group leader at the Max Planck Institute of Marine Microbiology, still in Bremen, and she's also a project leader and scientific head of the single-cell imaging facility. Her research is focused on anaerobic microbial metabolism, anaerobic respiration in prokaryotes, bacteria and archaea, as well as eukaryotic, protozoa, giant foraminiferal microbes. She's interested in microbial symbiosis among prokaryotes and microbial cycling of greenhouse gases. Please.
Yes, thank you for the introduction. Can you see my slides all right?
Yes, perfectly.
Okay, thank you. Yeah, thank you again. It's really a great pleasure to be able to be here today and talk to you all. maybe if it wasn't obvious enough from the introduction, I have to disclose that I am actually relatively new to the world of microeukaryotes and their symbionts. Me and my group actually work on free-living prokaryotes, Bacterial and Archaeal mainly. But during our research some time ago, we came across, quite accidentally, I must say, across an unusual endosymbiont of an anaerobic protist. And in the next 20 minutes, I would like to tell you a little bit more about how this discovery unraveled. So if you let me start with introducing you to our study site. This is Lake Zug. It's a freshwater lake in central Switzerland. We have been studying this lake for a few years because it is very interesting from the perspective of greenhouse gases, which is the research focus of my group. The lake is meromictic, which means that it doesn't turn over during the year. So the bottom waters of this lake remain permanently anoxic. It offers very stable conditions for anaerobic prokaryotic as well as eukaryotic life. Oxygen is depleted in the upper waters, and nitrate is present in the water column, and we know that it is removed in the anoxic hypolimnion through the activity of anaerobic prokaryotes mainly. We collect our waters from the deepest points of the lake, which is in the southern basin. It's around 200 meters deep. And we traditionally would use Niskin bottles to retrieve the water and prepare it on the boat. And our particular interest in the last years have moved on to nitrogen cycling and anaerobic nitrogen metabolism. We are on a search for microorganisms that are capable of anaerobically respiring nitrate. This process called denitrification is a four-step process in which nitrate is gradually reduced over formation of nitrite, nitric oxide, and nitrous oxide to the nitrogen gas. This process is very well understood enzymatically and biochemically in many prokaryotic models. And so we have the advantage that in addition to using biogeochemical analysis, we can also rely on molecular methods to a large extent to help us identify denitrifying microorganisms. And for me and my group of particular interest is this gene, that's the NeLLi gene that encodes for nitrous oxide reductase. And the particular interest in this gene is that it's to date the only enzyme that we know that is capable of removing nitrous oxide biologically. So our molecular approach to looking for nitrous oxide removers or denitrifiers was to look in which taxonomic, in which organisms do we retrieve nitrous oxide reductase gene. And for this, here it is depicted on the so-called GC coverage plot of a sample, metagenomic sample that we retrieved from the deep anoxic waters from 160 meters. And when my student, Jon Graaf, who was the lead author on this paper and who spearheaded this project, first saw this graph, we were sitting over it and we were wondering, there's this one contig right here that you see is sitting really far away from all the other contigs in the sample. And it contains the nitrous oxide reductase. Why it is so peculiar, why it is sitting so far away from all the other contigs is because it has a very low GC content. And originally we were not quite sure whether this is a real thing that we're looking at or some glitch. But when we compared to our corresponding metatranscriptome data, we saw that the gene, the NeLLi gene on this contig was actually very highly expressed. And of course, for us, high expression would be the first indication that maybe what we're looking at is a potentially very important and active member involved in the process that we're studying. To our luck, it turns out that the contig that contained this gene, despite the small size, as you can see, it is less than 500,000 base pairs large, is in fact a closed genome. And that, of course, opens the possibilities for studying or for elucidating who this genome belongs to and who is this putative nitrous oxide reducer. Some indications came out very fast after we looked at some basic properties of the genome. As you can see, the size is very, very small. It is less than 300,000 base pairs large. The GC content, of course, is very low. It only encodes 310 protein coding genes, which is very little, but it has a very high protein coding density. And all these are features of genomes of obligate endosymbionts. Obligate endosymbionts, so bacteria mainly that co-evolve with their hosts for large periods of time, tend to reduce their genomes dramatically through gene loss or through gene relocation to the host nucleus. And this process is often accompanied with a sharp increase in the AT content of the genome. And this is also, you can get a feeling for it here on this graph that Jon prepared, where he just pulled out genomes from the database, and the free-living prokaryotes are in gray, the background that you see when we move to a genome sizes that are less than 1 megabase pair. This decrease in genome size is accompanied in decrease in GC content and a stark overrepresentation of mainly insect endosymbiotic, but also protist endosymbiotic genomes. We did possess the whole genome with a complete 16S operon that allowed us to use the 16S RNA gene as a taxonomic marker to study the taxonomic position of this organism. And when we put it in a tree, we saw that the sequence falls into an own order within gamma proteobacteria. It has the highest similarity on a 16S to intracellular and intranuclear parasites from the Legionellas and Archaeal, but the similarity is very low. You see it's only around 86%. We called this organism Candidatus azaamicus, Ciliaticola, we call the whole clade Candidatus azaamicus. And if you look a bit closer, here you see that it is actually composed of 2 subgroups. There is one subgroup highlighted here in red that encompasses sequences that are very closely related to each other, high similarity to each other. And also, interestingly, these are all sequences that are retrieved from freshwater lakes, even though these lakes are geographically very distant from each other. We have lakes in Africa, Europe, and Asia. When we move to the group B, we see that the similarity on the 16S level drops, and also the heterogeneity of the environment from which the sequences are retrieved increases, where we're looking at marine samples, hypersaline samples, even wastewater. Based on the 16S sequence that we retrieved, we were lucky that we could design a specific oligonucleotide probe. The probe is coupled to a fluorescent marker and applied on a sample. It allows us to visualize the symbiont in the respective water sample. And the image from this fluorescent hybridization you can see here. The green signal comes from the fluorochrome coupled to the specific symbiont probe. And you see that the signal is localized in these discrete round spots located in larger organisms that are clearly eukaryotes. You can see the nucleus. You can even see the macro and the micronucleus here on DAPI. There seem to be 2 sort of morphotypes of the symbiont. Some of them, sometimes they are freely spread throughout the cytoplasm. Sometimes they seem to aggregate in these sort of conglomerates, but it appears to be clearly an intracellular and presumably cytoplasmic symbiont. This image was particularly important for us also for the reason that this was the first time that we actually had an indication of who the host of the symbiont might be. And at least we could see now that we are looking at a unicellular eukaryotic host that is around 20 micrometers large. And we could go back to our water sample and see whether we could retrieve or whether we see anything like that living and moving in the water in the lake. And indeed, this is how a drop of water with a picked one of these eukaryotes looks like. They move. They're very sensitive to oxygen and temperature, so they're hard to image. Normally they move much faster, and maybe you cannot see it very well on the video, but when we visualize them on the SEM, you can see that they're surrounded by ciliae. So this is a ciliate that we are looking at. And of course, to confirm that, we would perform PCR using general ciliate primers to amplify the 18S sequence of the ciliates to better understand the taxonomy. For that, we would handpick individual ciliates, and we would extract DNA from these single ciliates, or of course, for comparison, also from pooled ciliates. And we would perform double PCR. For each of these ciliates, we used endosymbiont specific primers to confirm that we're looking at the host that contains the endosymbiont. And we would use the ciliate primers to amplify the 18S from the picked eukaryotes. I must say, all the picked ciliates that we collected always contain the endosymbiont. And in fact, we know also from the hybridization studies that curiously, the bottom waters of the lake are nearly exclusively populated by the ciliate containing our endosymbiont. And when we look at the retrieved 18S sequences, we see that they nicely cluster together in one subgroup of a ciliate class called Plagiopelia. This class is maybe not so I don't know, known or popular. It's I think a relatively poorly described environmental group so far. There are some related cultured species that are presumably anaerobic and that are presumably relying on hydrogenosomal metabolism for energy. So knowing the taxonomic identity of the symbiont and of the host, of course, the big question was, how does the interaction between the symbiont and its host work? What is it based on? How does it function? And as we heard today, there is really two main functions for these kinds of endosymbionts in insects and in protists. And these are mainly serving either nutritional or defense capacities. For this, they tend to contain extensive biosynthetic pathways for synthesis, for example, amino acids or cofactors. In those 310 protein coding genes of Azomicus, we did not detect any of these biosynthetic machineries. I think we recovered only 1 or 2 genes for amino acid biosynthesis. So clearly, these traditional roles of symbionts are not what Azomicus is performing for its host. But if we look at the composition of the genes in the genome, we see that, for example, genes belonging to the TCA cycle are present. It is not a cycle in the symbiont because only selected genes are there, mainly the ones that pertain to the conversion of succinate and malate, for example, or that can generate used cofactors. We also have bacterial machinery for the synthesis of FES clusters and molybdopterin biosynthesis. Molybdopterin is an essential cofactor of nitrate reductase. We also have transporters for the molybdate and for the malate that can feed into the TCA and the biosynthetic pathway. We also found the SEC and the TAD pathway for protein transport. And of course, most intriguingly, we found electron transport chain in a composition where it only consists of complex 1, 2, and 3. Complex 4 was missing from the genome, and this is very peculiar because essentially all microorganisms, and there is many denitrifying microorganisms, but essentially all of them are facultative anaerobes. So they all have, in addition to denitrification, they also have capacity to use oxygen. In the Azomicus endosymbiont, the terminal oxidases were absent from the genome. We explored this a bit further because it was a peculiar observation. We actually performed transcriptomic sequencing of single ciliates to see whether maybe we detect transcripts for terminal oxidases, which was also not the case, or at least there was no clear evidence of that. And also we performed physiological experiment to show that the host, the holobiont of the ciliate with its endosymbiont, actually exhibit negative chemotaxis towards oxygen. The genome encoded complex 5, then again ATP synthase that can generate ATP from protein gradient, and a TLC transporter, which is a transporter that belongs to a family of antiporters that serve to exchange ATP and ADP across the membrane along concentration gradient. And of course, the genome encoded the denitrification respiratory chain. It encoded all 4 enzymes of the denitrification pathway, the NAR, NIR, NOR, and NOS. It also encoded the cytochromes and the accessory genes that are needed for the assembly of mainly the nitrate reductase. So that seemed to be very prominently conserved in this genome. So as I said, the nutritional and the defense capacity don't seem to be something that the symbiont is doing for its host. On the other hand, based on its genome composition, based on the selection of genes that it has retained over the course of the coevolution with its host, it seems rather that it has a role in the energy metabolism of the host. To confirm this, that this genomic potential is also expressed, we analyzed the metatranscriptomes from our samples, and there are 2 metatranscriptomic profiles shown here, and you see they're largely identical over the 2 investigated years that we show here. All the genes are shown, and the enzymes that I talked about before, such as the electron transport chain, the ATP synthase, the denitrifying genes here in blue, are highlighted as the highly expressed genes. And here I want to point out, this is the NOZ gene. And if you recall, this was the gene based on which we actually started paying attention to this genome at all. And indeed, you see it is the highest expressed gene in this symbiont genome. Looking at gene categories, we also see that any genes that are assigned to the category of energy production conversion, combined transcripts of these genes are composed more than half of all transcribed genes of the symbiont. With regard to denitrification, we went even a step further than transcriptomics because we have a handy method that allows us to actually detect denitrification activity even in small ciliates. And how we do is we use 59 labeled nitrate, and we monitor the conversion of the nitrate, the reduction to a dinitrogen gas, labeled dinitrogen gas. And you can see here, if we, from our sample, from our water, the lake water, if we remove anything that is larger than 10 micrometers, so basically all ciliates, if we remove the eukaryotes, we only have a background denitrification that can be assigned to the free-living prokaryotes. The moment we measure rates in the intact water, or if we measure rates in the water that was specifically enriched with eukaryotes, with ciliates, the denitrification rates increase. So providing a strong link between active denitrification and the ciliates in our sample. Based on all this combined genomic evidence, transcriptomic evidence, and also activity measurements and experimentation, we believe that azabicus is actually primed to provide energy in the form of ATP for its host. And this is a concept that we had to wrap our head around a little bit because, of course, everybody knows that in eukaryotes, these sort of core functions that we detect in the azabicus genome, such as respiration, such as ATP production from chemosynthesis, or even FES cluster biosynthesis, these are all functions that are traditionally performed by mitochondria. So there are specialized organelles in eukaryotic cells that are responsible for these processes. And in aerobic eukaryotes, these are mitochondria with aerobic respiratory chain. In anaerobic eukaryotes, these are mitochondria-related organelles that maybe rely more on fermentative types of metabolisms or substrate-level phosphorylation. Intriguingly enough, mitochondria too have evolved from a proteobacterial endosymbiont. Of course, this event happened much a longer time ago than the symbiosis that we are observing here. But it is, for me, an interesting thought to see which direction is the azoamicus ciliate symbiosis evolving, and whether we are maybe looking at a stage in an evolution of a symbiont that could give us any insights into the stages that maybe a mitochondrial predecessor has gone through as well. Clearly, azoamicus is evolutionarily unrelated to the mitochondrial line of descent, but it is really peculiar that during these, and we believe these might have been hundreds of millions of years of coevolution, the symbiont has selectively retained genes in its genome that are so functionally similar to mitochondria. And this is a sketch that we included in our paper, where we kind of speculate about which steps might have led to the establishment of the extant symbiosis that we observe now in the ciliate with the denitrifying symbiont and presumably remnant hydrogenosomes. We know that ciliates, just like all other eukaryotes, were originally all aerobes with mitochondria, and secondarily adapted to a life in anoxic habitats, accompanied by further reductive evolution of the mitochondria towards mainly, or most commonly, hydrogenosomes. And we believe at this stage, the ciliate has acquired the predecessor of the azoamicus and the symbiont. And this fork here is supposed to illustrate that we do not quite understand or know what was the genomic composition, or what was the content, the genomic capacity of the symbiont while it was still free living and acquired by the eukaryote. We know that ciliates are hotspots of horizontal gene transfer, and it is very likely that many of the genes that we now find in the symbiont genome might have been laterally acquired, or at least exchanged from the bacterial prey or other endosymbiotic bacteria. And yeah, with that, I come to the end of my talk. I would just like to wrap up. A last word about this symbiont. Azoamicus is and presumably organisms belonging to this clade seem to be obligate endosymbionts that confer their host the capacity to breathe nitrate. And they seem to be primed to be involved in the energy metabolism of their host by providing energy in the form of ATP. And this function surely expands the roles of these types of modern eukaryote bacteria symbioses that we are all interested in. And the big question for me is to understand what impact has the ecology, the environment in which this holobiont lives and evolves on the evolution of the endosymbiont. And particularly I'm interested, of course, with my background, I'm of course interested in the denitrifying genes. We see evidence that they have undergone lateral gene transfer, and it is really an interesting question to try to understand how this might have affected the establishment of this symbiosis. And yeah, with the last words, I would just really like to very cordially thank to my institute in Bremen and the whole Max Planck Society and our collaborators in Switzerland, with whom all this sampling was even possible, and the extensive sequencing work that was done, both metagenomic and metatranscriptomic, at the Genome Center in Cologne. And Jon, who has now moved on from scientific career, but who has really pushed this project very hard to a successful end. And Sina, who is now taking over this project to a large extent, together with a great support, bioinformatics support from Dan, our scientist. And yeah, thank you for your attention.
Thank you very much. We have a first question. do you have any estimate of the age of these endosymbiosis?
Yes, that's a great question. And we tried to include an estimate, but we are perfectly aware that this is very unclear. The estimate we made was based on the 16S, sequence, and we arrive at a couple of 100 million years, something around 200 to 300 thousand, 200 to 300 million years. Yes, but yes, it's a good question.
Okay. Yeah. Just one more curiosity. In the system, you don't have Archaeal Symbiont associated, isn't it? So no methanogens.
Yes, that was that is a good question. That was the first thing we were looking for. Indeed, we used Archaeal probe also to hybridize our Ciliate. We didn't get any signal. We also looked for a 420 autofluorescence in the Ciliate. We also didn't detect any. I should mention that the water, so the Ciliate seems to be planktonic, and we know that the bottom waters of this lake contain hardly any archaea. So it's very strongly bacteria-dominated system. Maybe that has something to do with it. Okay.
And do you expect to find the relatives of this group of bacteria in related hosts? Do you have any hint about it concerning the occurrence of 16S or 18S RNA genes in global metagenomic database?
Yeah, we had big hopes about finding a lot more about the biogeography and the distribution of the symbiont, and I can say that it is more widespread than this one particular lake in Switzerland, and it also seems to be more widespread than just confined to freshwater habitats. The question about the host is an interesting one, and unfortunately that is not so easy to find out. We are pursuing right now the approach that we are trying to retrieve the symbiont sequences and see if any of these environments we can go to and collect samples, because just retrieving from the database 18S sequences are not, doesn't seem to bring us much further. I think the ciliate physiology is not so well constrained in function as the 16S of the Endosymbiont.
Okay. We have one more question concerning your organism. It belongs to somehow Legionnalis, that is an infamous group of pathogen parasites. Is Azomycos the first mutualist that has been found in this group of intracellular microbes?
I must confess, I did not think about that this way. Right, that's a good point. I think we could probably look at it with more sequences available. Indeed, the lineages that are surrounding the Azomycos lineage are parasitic, yes. Also, the ATP-ADP transporter, in fact, also is sometimes present in parasites. So there seems to be some sort of a possible connection between a parasitic lifestyle. Who knows? Maybe it all started as a parasitism and only evolved into symbiosis later on.
By the way, the ADP-ADP transporter, did you try to make a phylogeny of only that gene to see if it has been horizontally transferred? Because if I remember correctly, it is present rather frequently in alpha proteobacteria, and especially in Cryptomonas and Holospiralis.
It is horizontally transferred. It is an alpha proteobacterial transporter. Yeah, you're completely right. Yeah, and that was also the sketch that I showed at the end. That was exactly to demonstrate this, that the ADP transporter was horizontally transferred, but probably many genes are not anymore the ones that were present in the original predecessor. It's of course hard. It's hard to treat them because we do not get clear answers. We get all these treeing artifacts. But with the ATP transporter, we're pretty sure that that one must have come either before or after it was acquired by the ciliate from a different proteobacterium. Yeah.
Okay, we have another question from the general question and answer session. Do you know of any free living Zoothamnicus relative?
No, I don't know. So you mean if there's any in that clade? Possibly. Hard to tell because the only thing we know about them are the sequences that we retrieve in their environment from which they were recovered. With the genomes that they have now, they cannot survive without a host. But yeah, which of the sister lineages, how far back one has to go with the sister lineages to arrive at a free living predecessor? That is an interesting question. Yeah.
I have an additional question for you. It's a bit of ciliate biologist. Your host is belonging to the Plagiopilea. That is a class of ciliates that contains only strictly anaerobic ciliates.
Yeah. Mm-hmm.
And Epaxella is in all molecular phylogenies the first split within Plagiopilea. And all other Plagiopilea that are sister to Epaxella are generally larger in size and are associated to methanogens.
Yes, yeah.
So it's
yes, with the hydrogenosome.
That is not common to the whole class, but just to aside one part of the class.
Yeah. Yes, we couldn't. It would be interesting to exchange any insight on this if you have, because we had a hard time retrieving much information about this group. So yeah.
Well, we have data on Plagiopilea, on Plagiopila, but not on Epaxella. We never found them, but we have Plagiopilea.
Yes. I wonder exactly, how much diversity is there with regard to their metabolism in these different clades of that group? Yeah.
Okay, Friedrich, I don't know if we shall move to the different rooms. How would you like to proceed?
Yeah, I think. Oh, Brooke, you want to?
Yeah, we could all proceed to the breakout rooms. I think Frederick already has them set up. So if you click the chat, the link is right there in the chat, and it should, if you click the link, seamlessly transfer you to the breakout room.
Okay.
But are there different rooms or just one room for?
There are different rooms. I'm not exactly sure which speakers he has in each room, but when you get to the rooms, you'll see a link at the bottom that says breakout rooms, and you'll be able to click which room you want to join.
So thank you very much for to the presenters and to you all for attending, and thank you, Giulio, for chairing the session. We hope to see you all next week for the last session, and see you in a moment in the breakout rooms.